Alright, so these are your biology notes for section 8.6 over gene expression and regulation. Your objectives for this section are to describe how prokaryotes turn genes on and off, and to explain how gene expression is regulated in eukaryotic cells. Alright, so our first thing that we're going to discuss here are operons. Right, so in prokaryotes, they can control the expression of a gene in a really simple way. They use these operons that include a promoter, an operator, and one or more structural genes. The promoter basically just attracts that RNA polymerase so that I can transcribe the DNA to make some RNA that's eventually going to make a protein. But the operon itself includes that promoter, but it also has a couple other things. It has an operator, and it's going to have one or more structural genes that are being controlled. Right? They're genes that you're turning on and off whenever you need them. So we have a couple different operons that we're going to deal with. The first one is this lac operon. That lac is short for lactose. So I need to draw a piece of DNA here, because that's where it would be located. And I have a promoter, which is a region that attracts that RNA polymerase. Again, I have an operator, which we'll see in a second how this thing works. That's actually how we turn on and off the genes that are being regulated. But over here, in this region, that's where you would find your genes that are being regulated. Okay, so operons work because there's a repressor protein that either will or won't attach to that operator at any given time. So this lac operon has this repressor protein that fits into the operator that turns it off, right? If that repressor protein is bound to the operator, RNA polymerase is blocked. Remember, I'm going to try and bring in that RNA polymerase over here where the promoter is. I'm going to try and work in this direction, and I'm going to transcribe some DNA. But if there's something physically in the way, it can't do anything. So that repressor protein binds to that operator to block RNA polymerase, to keep it from transcribing. This lac operon is said to be inducible. And inducible actually means that it's usually off, but if I need it, I can turn it on. So right now, this lac operon would be off because that repressor protein is attached to the operator blocking RNA polymerase. But if I need those genes over there to be expressed, I can turn this on. So the next idea here is where the lactose comes in. I'm going to change my repressor protein a little bit here. It's got another region on it that can interact with a different molecule. If lactose is available, and you guys know what lactose is, it's a sugar found in milk, right? But if lactose is available, then I'm going to need these genes that are being controlled here, because without expressing these genes and getting certain enzymes, I won't be able to break down that lactose to get any energy out of it. So what happens here is that lactose and that repressor combine, and when they combine, right? The circle fits into that circle slot. I'm going to see a change in that repressor. Right? Instead of having that triangle shape at the bottom so that it does fit into the operon, into the operator, there's now that square, that wedge shape instead. So it no longer can bind to the operator, and if it can't bind to the operator, that means that RNA polymerase is no longer blocked. I will be able to transcribe and translate that DNA over here. I should be able to make my lactose enzymes, ultimately. But you only need those lactose enzymes if lactose is available. So this system makes sense, right? If lactose, that green circle, is available, then I want to have lactose enzymes. But if there are no lactose enzymes, then I don't, or if there's no lactose available, then I don't need any lactose enzymes. So this is a reversible situation, right? Once lactose is not available, I go back to my repressor in this form. It'll bind to the operator and I'll stop producing lactose enzymes because I don't need those lactose enzymes if lactose is not available. But as soon as that lactose is available, that repressor is going to go into this form and then I will produce my lactose enzymes. So you can turn it on, but usually this is off because lactose actually isn't available very often for those bacteria. But when it does become available, they'd like to use it for energy, so they will express these genes. They'll make those lactose enzymes. All right, so that's not the only operon that we can deal with. We also have this TRP operon. That TRP is short for tryptophan, and tryptophan was one of those amino acids that we talked about. This one works a little bit differently. Similar to before, you still have a promoter region. You still have an operator. You still have some genes that are either going to be expressed or not expressed. So that blue region of DNA over there, that's where those important genes are. Those genes in this case are going to be used to produce tryptophan. Right, so I'm going to use these genes over here. I'm going to transcribe and translate them to produce that amino acid tryptophan that we talked about before, or that we've been using to build proteins, at least. So if you want to make more proteins, you're probably going to need that tryptophan. So as long as that stuff is needed in the cell, that those genes will be expressed, those enzymes will be made, we're going to make more and more tryptophan. But this operon is kind of the opposite of the other one. This operon is actually said to be repressible. Which means that it's actually usually on. Oops. But if you need to, you can turn it off. Or if necessary, you can turn it off. So the difference between this one and the last one is in the repressor protein. The repressor protein actually is normally in this form. Right? It's not going to bind to that operator because that wedge square shape doesn't fit into that triangle slot. But this time, if there's lots of that tryptophan available, enough of it that that tryptophan could bind with that repressor, that inactive repressor protein, I can activate that repressor protein because it'll change shapes when combined with that tryptophan molecule. And then that repressor will bind to the operator, which ceases the production of tryptophan. Right? This kind of situation, this kind of system works off of feedback. It's called feedback inhibition, right? If the whole goal of expressing those genes over here, those ones that are highlighted blue again, is to make lots and lots of tryptophan so that my cells can use those to make proteins, but eventually I've made so much tryptophan that I have lots of extra tryptophan. Once you have extra, it's a waste of energy to continue to produce it, which is why ultimately that system is repressed when there's too much tryptophan. So this system again, it's repressible. It's usually on, so usually the bacteria is going to make lots and lots of tryptophan. But once there's too much tryptophan, you turn it off. And again, it works by that repressor protein binding to the operator, blocking RNA polymerase, preventing transcription, which means that you can't translate that mRNA. You can't make the enzymes that make tryptophan. Remember, our lac operon was the opposite. It's inducible, which means that it's usually off. Right? Lactose isn't normally available for a bacteria to use. But if it is available, you want to have those enzymes to break it down to get the energy out of it. So this one's usually off. That operator is normally bound to the repressor, blocking RNA polymerase. But once lactose becomes available, this can turn on so you can make those lactose enzymes needed. So remember the difference between the two. One's usually off, you can turn it on when you need it. Lactose isn't normally available, but when it is available, you want to turn this system on. This one is repressible. The tryptophan operon is repressible because usually you need lots of tryptophan. But if you have excess, it's wasteful to make more. There's no reason to make more. So you'll shut it off when there's too much of it.